Okay. So last class, let's, I guess, get started. Last class, we were talking about gene finding. We're starting to talk about our unit on gene finding. And our problem is, again, I give you as input a, sequence of A, C, Gs, and Ts, okay, which is the genome of an organism. The output that you want to do is to tell me where are the genes on this sequence. Okay, that's basically what it is. But again, as good an accuracy as possible. Okay, any questions? And last class we talked about some of the properties of genes that might, how does this actually happen? Obviously there's got to be some machinery that copies it into RNA. It's got to correspond to the part of that RNA that corresponds to the triples that will describe how to make a protein. And we talked a little bit about the biology or the chemistry underlying that. But if we're going to write a program, in principle, the chemistry and the biology doesn't seem to help us too much because the exact way that this is done is somewhat is still somewhat unknown. But there are lots of features of genes that we can use to try to build a prediction model. And so what I'd like to do is to go through what are the features in genes that might make predicting them easy or reasonable. Okay? Any questions about where we are, what our goal is? Now, if you are charged with writing a program, this is what you would need to know. The first feature we tell you is that introns, the gaps between genes, okay, tend to be flanked by what they call donor and acceptor signals, AT and GT. Okay? So one way you might think of trying to identify where the intron boundaries are is look wherever there is an AT and say that's the start of an intron, and whenever you have a GT, that is the end of an intron. But why is that a bad idea? Okay? Excuse me, what? Well, one problem you're saying is that ATs might very well occur within genes, and that's certainly true, right? The main thing is that ATs and CG patterns will occur a lot of the time. Roughly 1 out of every 16 bases, okay, is going to be, what's the probability that starting at this point there is an AG? Well, if there's a 1 in 4 probability of an A, 1 in 4 probability of a G, every 16th position is going to be a donor site or candidate donor site, and every 16th position is going to be a donor acceptor site. So these are very weak signals. Does everybody agree with that? There's other properties of genes that might be interesting. One is from the coding sequence. Okay? So if we look at a coding sequence, every gene, every protein sequence, it turns out there's a start signal of ATG. Okay? That is the triple that corresponds to one of the amino acids, in particular it's the M. Okay, I think it's methionine. Okay? And that is at the start of every single protein. Okay? So one way that you might think about trying to do gene finding is look for where the methionine codon is, right? This ATG symbol. But what's the problem with that? Okay, if we have a system that says, okay, we'll look for where there is an ATG symbol, what is the problem with this?
ATG might also occur in the coding sequence.
ATG can certainly occur within the coding sequence. And more than that, by chance, it's not a very uncommon pattern. If I randomly put my finger in the genome, 1 out of every 64, with probably 1 out of 64, there is going to be an ATG, right? So we know that genes start with a start symbol, and they also end with a stop symbol. There are 3 possible stop triples, okay? So one way to think of a gene is it's going to be something that starts with an ATG and ends with a stop, but we see there's a lot of ATGs, and there's about a 1/20th probability, or 3 out of 61 probability, that when you start, put your finger someplace, the next 3 bases correspond to one of the triples. Okay? So these are weak signals. Any questions? Now, maybe if you combine enough weak signals, that will help you out. Does that make sense? What other stuff do we know about genes? Okay? One thing that's kind of an interesting statistic is that we know that if a gene starts with an ATG, ends with a stop, okay, and then there is every triple, a series of other triples. One thing we also know is that the distance between here and here is congruent to 0 mod 3. It's a multiple of 3. Does everybody agree with that? So this looks like a clue we might be able to use, okay? Except that, 1/3 of every sequences will be of multiple 3 in length. Does that make sense? And also, if we have exons, this clue is not very useful, right? If we have a world where our gene consists of coding an exon followed by an intron, again I said GT, AG, here would be the start, here would be the stop. The sum of the lengths is a multiple of 3. But when we split into exons, there's no reason it's splitting on, frame boundaries, okay? So this is a very weak signal, okay? Any questions? What other features do we have? It turns out that if you look at the statistics of how often what triples occur within genes, triples or sixmers, okay, occur within genes, they are slightly different statistics. There's a different probability distribution than junk, okay? So what am I saying now? If I have a real gene, okay, if you look in genes, okay, and you look outside, these are the exons, and you look at the introns or the, other non-coding regions. And if you take a look at all the species, then you count how often the probability distribution, the frequency with which, let's say, triples occur, they will occur differently within exons than outside the exons. I guess one way to think about it is, let's say that the exons corresponded to English words, and the stuff that was introns was gibberish, right? You could kind of imagine a world where the English words were written by people who know English, and the introns were written by monkeys, right? Monkeys would probably have a lot of symbols XCG, right? Where they banged on a typewriter, right? You've heard this thing, right? If a monkey was banging on a typewriter, you'd expect the pattern of triples of letters that the monkey types would be different than the pattern of triples the graduate student types. Does everybody get that sense? So what we could imagine doing is now build up the frequency for all the triples, let's say within a region, that we know corresponds to exons. Let's say we take a look at it. We have a set of genes that have been found by some method, right? Somebody has looked at it and said, this is a gene, and this part is not a gene, right? We can gather statistics based on what those are, right? And then by looking at the pattern of triples in a region, we can ask ourselves, is it more likely that it comes from the gene region or the non-gene region? Does that make sense? Okay? So if I see the word, if I see a window that looks like a, act space, this window probably, if it was in English, probably makes sense. That's a relatively high probability set of triples, let's say, in real English. Whereas if I saw ATGX, okay, that would be a low probability set of triples. Does that make sense? So you can imagine using this some kind of a frequency distribution of how often patterns occur within a region to tell me whether or not this region is likely in a gene or likely not in a gene. I feel I said that badly, but how many people see what I'm saying? Okay, I think that distinguishing between English and text by a monkey makes that example clear, right? Okay? Or if you wanted to get even more subtle, let's say English and Spanish. You imagine there's an English person and a Spanish person fighting for control of the keyboard, right? If you knew what the distribution in Spanish was, okay, they used letters at a somewhat different frequency than English, you would expect to maybe look at a window and say this text looks more like it's Spanish than English, right? Just by the basis of how often triples of letters come up, okay? And we can do a similar thing in gene prediction, okay, because there is a difference in the frequency distribution. The trouble is you need a relatively long window. If, let's say, you imagine there is a difference between let's say, three-letter distributions in Spanish and English, okay, you need a relatively long run for the statistical differences to show out. Does that kind of make sense? And again, the more similar the two distributions are, the longer the region you have before you'll be able to establish that with some confidence. Does that make sense? But now you could certainly imagine taking this kind of statistical, these triples, and doing something like the following. Okay? Here's my genome from the 5' end to the 3' end. I measure what is the probability of it being based on a window of the last 1,000 characters. What is the probability of it being a gene? Okay? You could imagine that if this was a gene, if I look at the statistical distribution, over here the probability of it being a gene should be relatively low, right? If I just take a window of 1,000, an basis ending in there, we would kind of expect that as I walk through this gene, the window is going to have a higher probability of being of having gene stuff, and then going down. Does that kind of make sense? So if you have a distribution, you might expect that these probability bumps might correspond to genes. Okay? Any questions about this? This is supposed to be clear and reasonable. And if it's not, then I'm saying it wrong, and I'd like to give it another try. Any questions? Okay? Yeah.
When you say there mostly multiples of 3, when exons are split,
Yeah.
are they close to 3 as in there will be 3 or, mostly 3, 6, 9?
No. So the trouble is split exons. When we have a split gene, the assembled final spliced product is going to be of length multiple of 3. But there's no reason why this might not have, sort of 3x plus 1 base, and this be 3y plus 2 bases, right? The sum would have to add up to be a multiple of 3. And there's nothing, the system doesn't seem to care at this point whether all the triples are in one exon or not.
My question is, what is the difference between an intron of length 4 and an exon of length 4?
So what is the difference? So the intra-exons are the stuff that will eventually be described as become part of the spliced RNA, will become part of the gene, right? The introns are the stuff between this, the gaps between the two parts of this gene, right? So the stuff that describes how to make the protein is this and this, okay? You would like to know that says, okay, in this region on the genome, there is a gene, and you'd like to know the part that makes the proteins are here and here. That's what you'd like to learn from your gene finder. Does that answer your question? Okay, any questions? Okay? So these are the features that I claim we get that will help make it reasonable to now start thinking about, yeah, question.
Is it not possible that all combinations of 3 in a codon, all 64 combinations map to protein bases?
Okay, so if we go back, there is a table which I think I showed you, during the first day of class, showing you that for every H, C, G, and T, right, the first base, there were 4 columns for the second base, and within it there were 4 other possibilities. For every triple of DNA, it will map to either, in the coding table, one of the 20 amino acids or a stop. 3 of them map to stops. The others map to amino acids. Okay? And that's the interpretation if it is considered a gene. Now note in the introns, there is also a sequence of stuff that in principle, properly interpreted, could be thought of as saying, oh, this would code for an amino acid if that was part of the gene. But that's somehow the system does not recognize that sequence as a gene. Any questions? So there's certain parts. It's what I'm saying is the following. Because there is a coding table mapping triples to amino acids, anything, even junk, can be interpreted as coding for amino acids. Is that right? What we care about is what are the places that really are biologically actively encoding proteins. And for a variety of reasons, only 2% of the genome is doing that, right? And our goal is to find that 2%. Any questions? Okay, so this is a sign that we have some features we can play with, right? And that is a good thing. Okay, let me just try this thing. Hold on a second. Now, there are several problems that make this harder, okay? We sort of now have the start of, let's say, a gene-finding methodology, right? There's certain functions, symbols that have to be there. They're weak. There are certain statistical differences within genes and not genes. We can look to see where the line goes up and down. Recognize that there's lots of problems that make this very hard. One problem turns out to be that the exons can be separated by several thousand bases. This is one of these things I find amazing, actually. So part of me says, okay, if exons are split, fine. You have an exon here, you have a little intron, okay? Somehow, the biological machinery might interpret it as you have several thousand bases here. And here is a little intron, okay, a little exon. And then thousands, tens of thousands of bases might happen before the next little piece of that protein comes on, right? And somehow the machinery is smart enough to splice all this stuff out, okay, and stick it together. But that means that we have to be aware that there might be long ranges where there is no exon, okay? Any questions about that? So that's a discouraging point. Another thing that's discouraging is that in a world where we're sequencing data, the kind of sequencing errors we get might very well cause problems. If we're looking explicitly for a GT as a donor site, if we happen to have a sequencing error, and that's an AT, by definition we will miss that exon. Is that clear? So you've got to deal with sequencing errors. Okay? And that's actually not as big a problem as you might think, because there are probably not that many in data these days. But we got to be aware of that potential. A bigger problem is that there are these things called pseudogenes. Okay? So one phenomena that I think I've mentioned in here is often in the course of evolution, let's say that you are a bacteria growing along. Maybe you have a genome that's doing its thing, right? And sometimes there is a gene duplication event. Did I mention these kind of things? Where suddenly, for some reason, a portion of the genome is copied and inserted into the organism again. So that would mean now I've got 2 copies of each gene, right? If this was gene X and this was gene Y, XY, after a copying operation like that, okay, a recombination event, I would now have 2 copies of the same genes, okay? And that's fine. But let's say the organism really only needs 1 to survive. Okay, and having the second copy of the X gene doesn't really help it. What we would expect to happen is somewhere along the line there would be a mutation knocking out, damaging this gene, right? It makes a slightly different protein. But because we didn't need that, we already are getting enough of that protein from here, the organism wouldn't notice it, right? You could kind of imagine that over time there's going to be a certain number of mutations, and the dead copy will drift away because we don't care about it very much, right? Does that make sense? But what's our problem now? If you now run a gene-finding program on it, this is a real gene. This is a statistically very, very similar sequence that represents a dead gene. Does that make sense? And so trying to tell which is the live gene and which is the dead gene is another problem that we have. Okay? Any questions about that? Okay? So this is a phenomenon we have to deal with. Okay? And finally, the last thing that makes modern gene finding very difficult. In modern gene finding, I'll say, is now we're starting to sequence mammals. Okay, we care about mammals because we're a mammal, right? Is that exons can often be assembled in many, many different ways. Okay? So we have this vision now, which I've been telling you about, that maybe there's this gene that requires the concatenation of these 4 exons. Does that make sense? But it turns out a slightly different protein could happen if we just took exons A, C, and G. Does that make sense? You could imagine if this was length that was a multiple of 3, you would get putatively functioning proteins if you assembled A, B, C, and D as a gene, or if you assembled A, C, and D as a gene. Does that make sense? And it seems that a lot of the complications, the thing that makes humans more complicated than other things with similar numbers of genes, is that there's more ways in which this alternate splicing can happen. Okay? And so what you'd really like to do from your gene finder is not just what are the pieces, but how are the ways that they should be put together, might be put together. Okay? Any questions about that? You'd really like to say, okay, here's a hunk of, you give me this hunk of genomic DNA, I tell you there's 4 exons, and in fact, the way that they are probably going to be assembled is A, B, C, D, or A, C, D. Any questions? Okay? So this is why the problem is hard. We're not going to deal with this particular problem of alternate splicing. We're still going to be dealing here with just the problem of recognizing the exons. Okay? What is an exon and what is not an exon? Any questions? Okay. Now, how do we know what is actually a gene or not? Okay, there are some laboratory ways, experimental ways you might find out whether something is a gene. Okay? Right now we have this world that this vision that we took as input, we sequenced the genome of a human, okay, which was DNA, right? And we want to find where the genes are on that. There are different ways in that we might be able to get some insight as to what is a gene by doing laboratory experiments. One that I think makes sense for us is, what if we could sequence RNA? DNA and RNA are very similar molecules. By copying the molecule properly, you can sequence RNA, okay, or a DNA copy of RNA. And so think what this might do. Maybe what we could do is something like this. Take a cell, extract out all the messenger RNA in the cell, and just sequence this stuff. Does that make sense? The DNA got copied into the RNA where there were genes, right? If we got copies of it, okay, and we just sequence this, this will tell us what is actually being transcribed. And that should be able to, by doing a blast search and aligning that on the genome, That should tell us where there are genes. Does that make sense? Okay? What's a limitation of this? Can anybody think of anything good or bad about this method? The good news, it would seem, is that only 2% of the genome is going to be genes, right? If I just sequence the RNA, that's coming from the useful 2%, right? So it would seem like that was a great shortcut, especially at the beginning. Why sequence all the junk? Why not just get the genes? Okay? Any questions or comments about that? So this looked like a great idea. It is a good idea. But there was a problem. The problem is, suppose you wanted to learn English by repeatedly sampling words. Let's say I want to learn all there is to know about English. Okay? And the way I'm going to do it is I'm going to randomly sample a word somebody uses in practice. Okay? And after I sample enough words, I will know all the words in English. Does that make sense? What's the problem with that method?
Certain words
Yeah.
you don't know. You wouldn't know how they relate to each other.
I wouldn't know how they relate to each other, but still, knowing a parts list of English would be useful. We're trying to build a parts list here of what are the exons that are expressed. Okay? So that's not a bad one. This one this was a better problem.
Certain words aren't spoken as frequently.
Certain words are spoken infrequently, and certain words are spoken frequently, right? So what would happen if this was spoken English? What would happen? Every fifth sample you get is the word the. Does everybody see that? And it will continue to be the word the, no matter how many times you sequence this. Does it make sense? You get 100 words from these people, 20 of them are going to be the, right? You say, no, I'll pay for 1,000. 200 of them are going to be the. Does that make sense? Now, the more words you sample and sample, you'll get rarer and rarer things. Does everybody agree with that? Like, what's an extremely rare word in English? Anti-disestablishmentarianism, right? Now, if you sample enough English, you will eventually see that one, right? But it will require a huge amount of sampling. And that's really what the problem is. That if you sample the sequenced stuff, okay, you end up getting a small number of genes that are expressed a lot. There's lots and lots of copies of them. And it's hard to find the other ones. And just because something does not appear that often doesn't mean it's not important. Some of you may say, oh, what's the? Must be pretty important if it's 20% of the word, right? But if I left out the word the from every time I said it in my lecture, it would not change the information content of my lecture very much. Does everybody agree with that? And a similar phenomena probably happens biologically. A lot of the important genes are not very heavily expressed. Any questions? Or they might only be expressed in a rare situation, right? So you can imagine some gene that only goes on, when the cell is exposed to a particular, let's say, heat shock or something like that. You might not see this under normal conditions. Any questions? So that's a problem with sampling all the RNA. Another question might be, is there a way of sequencing amino acid sequences directly? It turns out there are experimental approaches to that through something that's called mass spectrometry, which basically determines the weight of it takes molecules and it fragments them and then determines the weight of the parts, okay? And then given a vector of weights, you try to figure out what the amino acid sequence had to be. So these, it turns out, give you some information, but they're expensive, they're clumsy, they don't give you as much information. Much better for the biologist would be if we said we could just give our genome sequence to a computer scientist and say, have them tell us where the genes are. Any questions? And that's what motivates the pattern recognition problem. Any questions? Okay, good. So how did the first generation of gene prediction programs work? Okay? They basically did things like we were talking about. Okay? They took as input, Grail was sort of the favorite or an early favorite. Okay? It took as input a DNA sequence, a DNA sequence, it did things like identify all the donor and acceptor sites. Oh, here are all the places where there are donor sites. Here are all the places where there are acceptor sites. Here are all the places where there are ATGs. Here's all the places where there are stop symbols, right? It would then look within windows of the genome, maybe 1,000 bases long or 100 bases long, and see how does the probability of this sequence being within a coding region. Is it more likely to be in a coding region than not? Producing a graph, if we wish, of likelihood of being a gene versus non-likelihood. Okay? And now putting that all together with dynamic programming or whatever to score these things, you can now imagine how you might make a guess like this. Does that make sense? Okay? And based on these ad hoc features that we have discussed, we should be able to produce a gene finding program. Any questions? Now, I haven't yet talked about what is really the most important feature for these kind of gene-finding programs, which is the search for what we would call large open reading frames. Let's think about what, if we just think about a random sequence, okay, what is a reading frame? A way to think about the genomic DNA. Oops, sorry, what am I doing here? Uh-oh, did I lose my pen? That's a bad thing. Uh-oh, I'm in trouble if I've lost my pen. Did I lose my pen? Okay, let's give it a rest for a second. If we think about what's going to happen on, a random sequence, a non-coding sequence, let's say we have an ATG symbol corresponding to the start of a gene, right? And this was just, let's say, a random sequence. How long would we expect to go in terms of triples until we hit a stop codon? Okay? How long would, if let's say we don't worry about split genes, we just worry about genes. If genes were, a contiguous exon gene, so it has this beginning, it has an end, how often in a random sequence would we expect to go before we hit a stop codon? Or a symbol for a stop codon? 164. The argument would be that this triple here would have a 3 in 64 chance, right? Because there's 3 stop codons, or roughly 1 in 20, right? 1 in 20, 1 in 20, 1 in 20, right? We would expect that something like about 20-ish, okay, triples, or about a space of about 60 is where we would expect to see the next stop codon. Does everybody get that idea? Now note that this has to do with how you read it in phase. One interesting phenomena of DNA, just to make sure you're understanding this. If I start reading the thing offset by 1 base, does it seem clear that over here is another interpretation of a gene? If a gene starts, in principle, starts at a position and goes every triple, right? It might very well be the case that there be lots of stop codons here, right, in the alternate reading frame. That has no impact at all on this particular interpretation of the gene. Does that make sense, what I'm saying? Okay? Just like what pairs of letters are next to each other only make a difference in words. If we stuck words together, we don't care about the pattern of letters ending one word and beginning another one, right? So what is the point? The interesting thing is that if we start at a random place and keep going until we hit a stop codon, we would expect on average it will be about 60 bases long. Does everybody agree with that? Now it turns out that genes on average are much longer than that. Okay? An average gene is about, I'm just going to make up a number, let's say 200 triples long. Okay? It is very unlikely that we could go a stretch of 200 triples without ever hitting a 1 out of 20. Does that make sense? Sort of like playing Russian roulette with a gun that has 20 possible chambers, right? I put the bullet in one of the chambers. For me to get 200 stretches long, I've got to not hit the bullet every time. Does everybody see that? And the probability of that happening for something of length 200 is vanishingly low. Does that make sense? So if we see a long, what we call open reading frame, a long spot on the genome where there is no triple, no stop symbol, that probably did not happen by chance. And it is probably the result of evolution, so some form of selection, and it probably represents a gene. Does that make sense? Any questions about that? So one problem is, maybe the single most important feature is to look for long open reading frames, okay, where there's no stop codon, and say that that is a gene. Any questions? This actually suggests something about how genes evolved, right? I kind of imagine that at the beginning there were very short exons, right? To make a protein much longer, what probably happened? You could kind of imagine that there is a mutation event that comes out that changes this amino acid, this triple, from a stop symbol to a base, right? That's going to add to the end of this RNA till the next stop triple. Does that make sense? Now the old protein is going to carry along a tail with some other thing. Maybe this is detrimental. If so, the organism dies, and that's the last we see of it, right? Maybe it is neutral, okay? In which case the organism doesn't care if this extra protein is stuck on. But now there is room for other evolution that can happen here, and maybe it'll evolve a function. Or perhaps knocking this thing out, there was an extra advantage to having this tail. Maybe it bound better to its target or something like that. And so we kind of imagine that through this process, exons got longer. Okay? And so a long open reading frame is a good way to detect the gene. Any questions? Okay? So this is very good if you want to write a simple gene finding program. It means that it's very, very easy to find long exons. The trouble is some exons are very short. And of course, those are the ones we'd like to try to figure out. Okay? Any questions about that? Okay? So this is one way we might think about building a gene finding program. Another way we might think about building a gene finding program has to do with homology. Okay? We have this vision right now that there is a common, if you have two species with a common ancestor, at one point the common ancestor had a particular genomic sequence, right? Over time, things evolved. You can imagine that, let's say there was a common ancestor, let's say to a mouse and a human, right? Maybe it's been, I don't know, 100 million years since we've had a common ancestor. That's long enough that the stuff that is junk, that really did not have a function in either species, probably absorbed a large number of mutations. Does that make sense? But we have essentially the same genes as mice, okay? So you would imagine that if there was a critical gene in humans, okay, it's probably also a critical, has a homologue that is critical in mice, right? Does that make sense? So if I took a look at what I would expect the genome of human and mice to look like, I would expect regions where they were in common, okay, probably very similar, probably are genes, because a change in one of these critical things probably is detrimental, right? If you have a working program, I randomly change a base in it. I might improve the program. But more likely, I'm going to make it worse, right? So you would expect that any regions that are conserved between the two species probably represent genes. Does that make sense? So now if I give you the genomes of two other organisms, okay, I claim that by doing alignment of them and just looking for high-scoring regions, that might help me identify where the genes are. Does that make sense? So homology is a good way to do this, some of this kind of thing, and is one of the reasons why we have databases like BLAST for people to look similarities, and why a certain class of gene-finding programs basically work by cheating. They said, where in the genome are there where in the database are there other genes people found? Where is there something homologous to this? That is probably an interesting thing. Any questions? Yes?
So, we consider that the junk always have a uniformly random distribution.
So the question is the junk always have a uniformly random distribution? It's probably not a uniformly random distribution. There's no reason why we should think of it. But we should expect that the distribution is going to be different than within coding sequences. And more than that, if it doesn't have a strong function, it's more likely to change over time than the stuff that did have a function. That's the real critical principle here. The stuff that it was important didn't change. It's going to be the same between the two species. Okay? And the claim is that if you look at two species that were far enough away, the stuff that was conserved is probably important. And a large class of important things are the protein coding genes. So they certainly give you insight about where to look. Any questions? In fact, the way that people really identify the complete boundaries of organisms, of genes, the way that eventually people will really identify the absolute boundaries of genes is by aligning many different species together. So there was a very interesting study done where a group, I think it was MIT, sequenced 4 different strains of yeast. To me and you, yeast is yeast, right? But there are lots of different yeast species, okay, that are 20 million years apart, 5 million years apart, 100 million years apart, right? You could imagine a world where if you sequence all of them and you align them, if there is an important gene, okay, it's going to show up in all 4 species. And what's more, if it's, the difference has not been too great, okay, you should be able to use the fact that you have multiple species, every pair of which should have a homologous gene, to really pin down where the boundaries of the thing are. And so the most accurate laboratory way to try to identify accurate gene boundaries and eliminate these things that are dead genes is to look at several closely related species, look for very similar things, and do the right reasoning on them about what the differences are. Does that make sense? Okay. Any questions about that? Yes?
How similar do these organisms have to be?
How similar do they have to be? What you would like it to be the case is that they're similar enough that you can recognize when something is the same, but different enough that you can recognize when there is a difference between something that should be conserved and something that is not conserved, right? So if I want to find what are important genes in humans, it would probably not do to me too much good to sequence everybody in the room. Why is that? Because yes, your genes are going to be very similar, but also your non-genes are going to be very similar, right? There's not that much difference between here, right? But if I take a gorilla, okay, a gorilla diverged 10 million years ago or something like this. The proteins in gorilla are probably very similar to the proteins in human. There's been time for the other stuff to accumulate random mutations, okay? And now I can hope to look for high similarity regions to tease out that these things are higher similarity than the background, they're probably important. Does that make sense? Any questions about that? Okay. Any questions? Okay. Okay, and finally, there is a what I'd like to spend the next couple of classes talking about today and the rest of today and tomorrow. Talk about there is another class, sort of a second generation. So all these experimental procedures and alignments and stuff like that are good, but they require extra experiments. They require extra data. We as computer scientists like the idea if you give us the data, we will make a prediction as to what the problem is. Okay? Obviously, it's cheaper for us to just run a sequence through a program than do more experiments. So in principle, if we can build accurate gene-finding programs, this would be a good thing. Okay? And the first generation of programs was useful, but they still weren't that accurate. Okay? And the second generation of gene-finding programs, based on something called hidden Markov models, turned out to be a lot more accurate. And we're going to talk about that technology now. Any questions? Actually, can anybody think of a weakness in the homology system before I go any further? Trying to identify genes between, by doing this sequence comparison, one genome to another. What kind of genes will that fail on? Okay? Genes which are unique to a species, right? So you could imagine, for example, if let's say there's a certain type of, fish that lives very well in cold water, right? What is it, that lives in freezing water? It somehow evolves some form of antifreeze, so it's perfectly happy living in very cold water, right? If you do the sequence, the homology between the antifreeze fish and a normal fish, right? This will not show up as similarity. Does that make sense? Whereas, in some sense, what you're going to learn by doing these alignments are the genes that are like the ones that you already know about, right? That's good, but probably the real money is what makes something different rather than what is it that's the same. Any questions? And so you can't expect to do that so well from homology, okay? You may think you need to do, you need some other way to do sort of de novo prediction, okay? And that's where these hidden Markov models come in. Any questions about gene finding so far? Okay? This may be more biology than people wanted to see. Yes? Sequence proteins, yes.
But then it's somewhere that the code is degenerated so that multiple codons may code for one particular base in the protein, multiple triplets.
Okay, so the question is, what about protein sequencing? If I did protein sequencing, and suppose I sequence, and I get that this is my protein sequence. Could I find where my amino acid sequence of a protein? Would it be hard to go into a DNA sequence, a genomic DNA sequence, and find where the gene corresponding to this protein is? Is that hard, or trivial, or in between? It's not trivial, because each one of these symbols may correspond to up to 6 different triples. Does everybody see that? So it's not like I can translate this into a one-to-one, into an amino acid sequence, right? I mean, translate the amino acid sequence into a DNA sequence, and then look, is that DNA sequence here, right? So it's not trivial. But would it be hard? Not really. One way I might be able to do it that's kind of interesting is, what if I take the entire genome and interpret it as amino acids, right? I start from the first character of my amino acids, of my genome, and start reading triples off. And that will give me an interpretation in amino acid world, right? You could imagine I do the same thing 1 base over. You can imagine I do another base thing 2 bases over, right? Now, if this amino acid sequence is coded for by a gene in here, in one of my 3 frames, this sequence is going to exist. Does everybody get that idea? Yes? Okay, so this has a problem that you're telling me, gosh, introns aren't important, right? Are important. And if I'm looking for this exact sequence, if there was an intron in it, then I would only expect to see a large hunk of my sequence there, right? How should I get around that problem? Something like the Smith-Waterman algorithm, right? Wouldn't that be the right thing? Now if I do the Smith-Waterman algorithm between my amino acid sequence and my 3 translated amino acid sequences, what would I expect to see? For each of the exons, maybe here's one exon, here's another one, maybe here's another one in a different reading frame, right? I would expect to see high-scoring regions, okay, in my genome, right there, okay? So that's how I would do this kind of comparison. Does that make sense? Any questions? Okay. So let's start looking at, any other questions about this before I go on? So let's look at the HMM idea for gene prediction. And the idea behind this is a little bit different than, let's say, how I might normally have designed things. We first started talking about doing gene prediction. How would we have approached it? The basic way we approached it was ask our biologist, what is the properties of genes and what is the properties of non-genes? Oh, you've got these donor and acceptor sites. What do they start with? Oh, does it start with an ATG and with a stop codon? And then based on these features, we came up with an idea of an ad hoc way to do gene prediction, right? Another way would be to go to the biologist and say, I don't want you to tell me about genes. I want you to go give me a list of things. You tell me, show me what the difference is. Here are a bunch of sequences. Give me a bunch of sequences that are genes. Give me a bunch of sequences that are not genes, right? These are things that you ask your biologist for. And then they say, write a program that separates, given another sequence, decides whether it is closer to that or closer to this. Does that make sense? It's something that takes a bunch of examples of genes and non-genes and tries to learn from them. Okay? So this is basically the idea of using a learning approach. Okay? You start with positive and negative examples, and then have the program figure out what the right sequences are, what the right features are that separate genes from non-genes. And what's interesting is these learning-based approaches are often surprisingly good. Okay, maybe it's not so surprising these days. You guys live in a Google world, right? Why is Google so good? Ask any question to Google. It's going to give you the answer just because there's so much data in Google, right? And maybe this isn't so surprising. But as an algorithms person who grew up wanting to design specialized algorithms for a particular problem using my brain, the idea that you can build recognition programs that basically learn from data and examples instead of from understanding the problem is an interesting, is a different mindset. Does everybody get that idea? So in the early days of computational biology, there were the algorithms people like me, there were the learning people. We made sure we didn't talk to each other. Okay? As time went by, these people started talking to each other, and the methods that are now used are sort of a combination of both ideas. Okay? Which is, I guess, what's supposed to happen. Any questions? But you see you understand the basic difference here between a learning method and an ad hoc or a problem-specific method. And hidden Markov models are an example of a way to build these learning methods that has proven very, very important in gene prediction and other problems. Any questions? And this is what I'd really like to talk about in this unit as the technology basis. Okay? Any questions? So to do this, I'd like to talk about an example. I'm not going to start from now on the idea of using hidden Markov models for things like gene prediction. What do I mean by things like gene prediction? The input is I give you a bunch of your basic problem with gene prediction is I give you a sequence, and I ask you gene or no gene. That's really the question that you're asking about, right? So the problem is, we're going to assume a situation where you can give me positive examples that are genes and negative examples that are not. Okay? And the goal is, given a new sequence, which class does it fall into? Right? So we're going to talk about this kind of approach using an example that is fairly, that's sort of an easier example to talk about than gene prediction. Okay? So I'm now going to shift gears and talk about recognizing something called the CG island. This is from an example from a book, the book I recommend on the reading list by Endy et al. On hidden Markov models. I also put a link to some lecture notes, okay, on my webpage on hidden Markov models that cover these examples. So I encourage everybody to go to my webpage and look for the hidden Markov model lecture notes, not just my lecture notes, but these other ones I lifted from, I think it was MIT. Okay? That'll, I think, explain what we're doing a little bit better. Okay? But I'd like to now talk about a problem where, again, just like gene prediction, we imagine a world where we're going to be talking about identifying what some things are sequences of interest, some things are not sequences of interest. You give me a bunch of training examples, and I'd like to try to separate them. And in the application that we're going to talk about, instead of separating exons from non-exons, we're going to talk about something called CG islands. What is a CG island? It turns out that if you look at genomic sequences, there is often the case where there are a lot more CGs than you would expect. CG is a C followed by a G, right? How often should a C be followed by a G? Roughly 1 out of every 4 times 4, or 1 out of every 16 places, right? It turns out that there are in certain genomes, there are regions where there are a lot more CGs than exist elsewhere in the genome. Okay? And this turns out to be interesting for certain biological reasons. Okay? Just again, basically that it turns out that there are, for a variety of reasons, CGs are likely to be mutated to TGs. So if you look at the genome, there's less, in general, CGs than you would expect by chance. So if there is a region where there are more CGs than you would expect, that is probably a region. They're there for a reason. And detecting these regions is interesting because they focus biologists' attention on something. So for now, let's just say we want to detect these things that are called CG islands. Okay? And our goal is, given as input a genome, identify where are the places that are real, what are the boundaries of what are most likely to be CG-enriched regions, CG islands. Any questions? Now, if I were given this task, me as the algorithms person, what I would probably say is, oh, biologist, you want me to find CG islands? You mean things with a lot of CGs, right? Yeah, I mean a lot of CGs. Okay? So I might do an algorithmic thing and look where all the CGs are, right? CG, 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 right? And you could imagine doing a ad hoc thing, okay, where I look for regions, windows with lots of CGs in it, right? And I report back to the biologist, hey, look, here's a region with a lot of CGs. That would be the way I might proceed on it. But there's some problems with that method. One is, it's not clear when somebody tells me that they have a thing called a CG island, there might be other properties of this thing, other than that it has a lot of CGs that are also important. Does everybody agree with that? It's sort of like, if I were to look around the room, okay? I could imagine fixating on some feature of people, okay? And I say, okay, what makes a boy different than a girl, okay? And someone says, well, girls have long hair, okay? And so you could imagine then I'd write a boy versus girl detector that basically identified everybody with long hair, okay? And that would be accurate on some level, but it's missing the point. Does everybody see that? It's another thing that separates girls from boys, right? And if you're just given sort of an operational definition, unless that person who gave you the operational definition really gave you the right one, your program is not going to really do the proper distinguishing. Does everybody get that idea? So what is another way to build a CG island detector? One is to try to build something that will take a lot of examples of what biologists tell you are CG islands, and a lot of things that the biologists will tell you are not CG islands, and try to then learn from that and identify based on the training examples. Okay? And that's the approach that we're going to talk about from now on. Any questions about that? Okay? So now to try to separate boys from girls, you give me examples of girls here, you give me examples of boys here, and now I try to identify what features are important, and maybe long hair is partially important, but maybe there are other features that are important also. Okay? And if I can figure them out, I will have a better detector. Any questions? Okay. So hidden Markov models are basically a model that is built on top of a simpler mathematical model called a Markov model, or a Markov chain, or a Markov process. Okay? A Markov chain is a network of states where there is a probability of transition between each pair of states. Okay? So we can kind of imagine this as being a graph or a network where each vertex is a state, and there is probability of a coin flip changing our state. So one example might be, suppose, let's say, we wanted to build a machine that read a sequence of coin flips and tried to tell us whether we had seen an even number of heads or an odd number of heads. Does everybody see that idea? We could imagine building a machine with two states. This is going to be the even state. This is going to be the odd state, right? If I am in even number of head state and I get a head, I will go to the odd number of head states, right? If I am in the odd number of head states and I get a head, I will go to the even number, right? On a tail, I will stay where I am. Does everybody see that? So this is sort of what I would consider to be a model that is formed by a network of states. Each state is going to encode something about what has happened before. Here, what is before is I had an even number of heads or an odd number of heads, right? It doesn't encode everything that happened before. If I'm in the even head state, I can't tell whether I've had 100 heads or 50 heads. Does everybody see that? But it encodes a partial memory of what has happened. A short-term memory, basically. A partial memory of what has happened. Okay? If I assign probabilities to these transitions, okay, I can start to figure out what is the probability of being in a particular state. Suppose, let's say, I use a loaded coin, where heads is 0.7%, 70%, and tails is 0.3%. Now, if I know what state I started with, let's say at this beginning there were 0 heads, right? My claim is that for any sequence of coin flips, there is a probability that this sequence of coin flips happened, right? In this case, it would be what? It would be 0.7 times 0.7 times 0.3 times 0.7. Does everybody get that idea? And if we assume, let's say, that our strings are being generated by a, where there is a certain type of coin flip at a particular state, we can now start to talk about, for any sequence of flips or of observations, what was the probability that that sequence was generated? That's what's these kind of models are used for. Any questions? And you could imagine a world where the probabilities are different. Let's say that when we're in the odd state, we use a different coin than in the even state. Maybe we use a fair coin in the odd state. Does everybody see that? Then the probability of this coin flip sequence is going to be different. Does everybody agree with that? Any questions? So what is a Markov model? A Markov model is governed by a network of states, like the even-odd thing. The probability of being in a particular state at a particular time is completely dependent upon two things. It's dependent upon what was the probability I was in each state at the previous time step, and what is the probability, transition probability, from one state to another. Okay? This transition property thing is a property of the model, right? The transition probabilities here are the 0.7, 3, 0.7. I think I had a 0.7 here, right? Okay. The transition probabilities are the probabilities of the model. That is something that is fixed as a result of the model. Okay? The model is a network of states. There's a state transition function giving the probability of going from one to another. The probability for a particular sequence, like let's say this one, the probability of this sequence being in a particular state at a particular time is a function of what state was I in at this time, one step before it, and the probability, transition probabilities from that state to the next state. Any questions about that? Okay? So what's interesting about Markov models is Markov models permit us to record some knowledge about what has happened in the past. But it doesn't record what the path was that got us there. Does everybody agree with that? So to a certain extent, it's like this class right now. You imagine all of you have had a variety of paths to get you to Stony Brook here, right? But now what happens to you is not a function if this was a Markov-like model. Your performance does not depend upon how you got here. It only depends upon the fact that you got here, right? And so that's what we mean by a Markov model. Any questions? Okay? So what is good? Typically, okay, a character is associated with each state transition. Okay, like a coin flip moves us from one state to another. If we're reading a DNA sequence and running it through an automata, the next base moves us from one state to another state. Does everybody get that idea? And the idea is that this means that once you have a model, there is an association between strings of states and model and paths through the model. Any questions about that? So the transition probabilities are independent from a state, are independent of the path that took us there. Therefore, if we want to think what is the probability of a given string, it is simply the probability, the product of the probabilities of each transition. Okay? And so if I give you a model, certain probabilities, certain strings are going to be more likely to be generated by that model than other ones are, right? In the one I gave you before, it looked like heads had a 0.7 chance of being generated, where tails were 0.3, right? if I give you a string like T, 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 it would have a lower probability of being generated than an analogous sequence of heads. Okay? Any questions? And so what we're really going to be interested in doing is, given a string, interpreting it as a probability, what was the likelihood it was generated from our model? Any questions? The key thing that we're going to be doing here is multiplying a lot of probabilities together. One trick to remember is what happens is a problem. If I get in a computer and I ask you to multiply a bunch of probabilities together, it becomes very, very small very, very quickly. Does everybody agree with that? That if I take a look at 0.1 and I raise it to the 100th, okay, it becomes very, very small very quickly, and it quickly goes out of the bounds of precision of a computer, right? You start having to use exponential notation and stuff like that, and you lose a lot of precision. An alternate way to represent probabilities that are very small. If I have something that's the product of probabilities, because I know that the log of x times y is equal to the log of x plus the log of y. Does everybody remember that? If I want to multiply a lot of probabilities together, it's sort of an analogous way to think of it is just to sum up the logs of the probabilities. Does everybody agree with that? If I take the anti-log of the sum of the logarithms of these things, I end up getting the product, right? And so it's often useful to talk in terms of summing up logarithms. So if you hang around the hidden Markov model space, you'll hear people talk about log odds ratios and things like that. Logarithms we're going to use sometimes because multiplying probabilities is numerically less stable than summing up logs. And if I want to figure out if my probability is better than yours, if I have the log of my probability is greater than the log of your probability, then my probability is greater than yours. Does everybody get that idea? Okay, any questions? Actually, maybe I'll come back here. Let's look now at how we might build a Markov model for recognizing the difference between the CG islands and the non-CG islands. Okay? So let's look at a real example here. One thing we might imagine as a simple model might be something like this. Suppose we assume that here we have a model of 4 states, okay, where each state corresponds to the last sequence, the base scene. Maybe we'll just, as a routine, pick this one as being the starting state, right? If I now this line, this edge from here to here, means that given an A, my next character is a C. Does everybody see that? You could imagine a world where if I gave you a bunch of things that are CG islands, okay? Here are things that are CG islands, okay? How might I be able to use these positive examples to determine the probabilities in this simple model? This model has 4 states in it. How many edges does it have? Okay. 6, well, no. 6, from every base, there's 4 possible ways to follow it. Is that right? So that tells me that for each of the 4, there are 16 possible probabilities here, right? 4 of them are, if I have an A, what's the probability I get another a C, a G, or a T? Does everybody agree with that? How would I figure out what these probabilities are? Given the examples, the answer is count. Does everybody agree with that? If you give me a bunch of positive examples, There's going to be a bunch of A's in there. I can compute statistics based on how frequent each pair AX is, right? Each pair CYX is GXTX, right? And based on that, I can now come up with a transition probability from every state to what is the probability of going to the next state, from just looking at my CG Island examples. Does everybody agree with that? And you can imagine, if you give me a bunch of negative examples, I could do the exact same thing for the negative examples. Does everybody agree with that? I compute the transition probabilities for each of my negative examples. And I could build a separate set automata on 4 states, showing what is the probability of each transition. Okay? Any questions? In this case, since CG islands are supposed to keep their CGs, and they're supposed to be depleted in other sequences, we would probably expect that this edge, the probability of weighting on that edge, is probably going to be a lot different between the two of them. And it turned out that in the examples that they had talked about in the book, the probability of going from a C to a G was 0.27, or almost a quarter, in the CG island model, but only 7%, okay, in the negative model. Does everybody see that? But there might have been other probability differences here. So given this automata, how can we now try to think about, if I give you another sequence, what is the sequence that it, whether it's a CG island or not, yeah?
This calculate the probability, given this transition probability and the other one.
The way to do it is to take my new sequence, ATC, and go A, what's the product of this, times that, times that, times this, times that, times that, times this, times this. Does everybody agree with it? This sequence will have a probability of having been generated by this Markov model. It will have a probability of having been generated by this Markov model. A reasonable way to classify it is to say which one has the higher probability. Does that make sense? And that way I could tell what is a this gives me now a classifier for what is a CG island and what isn't a CG island. Any questions about that?
What are those dotted lines?
Pay no attention to dotted lines so far. Dotted lines will be important. Okay? We are only dealing with Markov models. Okay? So now what I've done is build 2 separate independent automata. One for the positive examples and one for the negative examples. Okay? Questions?
The entire sequence will be a mix of these two, right?
So this is, the concern is, what happens if my sequence is a mix of these two? Does everybody see that this is a problem? Now, if I give you a sequence, it's one thing if you give me a sequence and it either is a Markov, a CG island or a non-CG island. Then I would expect this method to be able to tell, right? If, on the other hand, it's a mix of CG islands and non-CG islands, what would I expect? The probability scores out, if this is where the CG island is, these probability scores in this region should be lower here, more common here, higher probability here than here. And outside this, the probability sequences will be higher here than there. Does everybody see this? So this idea of 2 separate models seems good if it's just to tell the difference between me and you. But if we have something that is transitioning between the 2 of them, this does not seem so good anymore. Does everybody get that idea?
But we
Yeah.
can do what we were doing earlier, some window length which passes over.
So one way you might think about how can we get around this problem, okay? One possibility might be you take your input sequence and you break it into windows, right? And you score each window. Is it better in the positive or in the negative one, right? And you might imagine that maybe it's going to score better as positive, 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 Well, this is going to score better as negative, negative, negative. Then this is going to score better as positive, right? So you could think about doing it in Windows. But what do you need to know to do it in Windows? What is the size of a window where you expect the difference to be significant, right? Are CG islands big things or little things? Okay? How much would you expect to tell? So in order to try to tell mixed sequences and to parse a sequence where it's a mix of in and out, like CG island, non-CG island, or exon, non-exon, we need a way to integrate those models. Does everybody get that idea? Okay? And that is why we're going to want to talk about what we will call a hidden Markov model. Okay? Which is going to be, basically, at least one way to think